Dear participants and colleagues, I will now present for about 10 minutes on coconut diversity, phenotypic versus genetic, and ex situ versus in situ conservation.
As the purpose of this seminar is training, we will begin by defining the scientific terms we need for discussing. The glossary will be presented on 3 slides. However, we have alternated the glossary slides with other topics to make the presentation more enjoyable. We will talk about phenotype and genotype, and the response of coconut palms to environmental variations. We will end by comparing and presenting in situ conservation and ex situ conservation.
So here is the glossary, on 3 slides. A gene bank is a type of biorepository which preserves genetic material. In the case of coconut palm, in 2023, all are presently field gene banks conserving accessions of living coconut palms. An accession is defined as a collection of plant material from a particular location, received by a gene bank to ensure sustainable conservation of a single specific cultivar, landrace, or population. In situ, this is when a species is located or conserved in its usual situation, in farmers' fields, in protected areas or sometimes in tourist sites. Ex situ, this is when a species is conserved outside of its usual location, such as in field gene banks as living trees or in vitro collections of tissues and embryos. In vitro is used when plant material is located or conserved in a glass receptacle such as test tubes, transiently or as a part of ex situ conservation. Cryoconservation means frozen in liquid nitrogen for purpose of conservation, at -196 degrees Celsius. The coconut palm is very sensitive to environmental variations. To illustrate this subject, I will take 2 examples. The first is that of the Madang brown dwarf in Papua New Guinea. The Madang brown dwarf is autogamous and genetically very homogeneous. In Papua it produces rather large fruits, of good composition, and with round nuts. We have introduced it into the Ivory Coast collection. Curiously, in Africa, it began to produce many small, elongated fruits of less good composition. In Ivory Coast, this collection plot was planted on quaternary sands. These coconut trees were planted with care, using the nursery plastic bag technique. They received abundant but standard manure. However, this plot had been cultivated in the traditional way with cassava, which had gradually exhausted the soil. A drought occurred about 2 years after planting. Then the coconut trees never recovered. Then, even with abundant manure, they continued to produce many fruits, but these fruits were small and of poor composition. Finally, from the Ivory Coast gene bank, we sent the Madang brown back to Vanuatu, where it was planted on rich volcanic soils. In Vanuatu, we then observed an even better fruit composition than in Papua. Yet it is the same genotype, planted in different environmental conditions. The coconut tree can often be compared to a human being. Imagine that a child is brought up in poor conditions, that for 10 years, his food is insufficient and generates many deficiencies and developmental delays. Even if the situation then improves, the child will have accumulated negative characteristics of growth and development that abundant food can never erase. It is the same for the coconut tree. A proverb says, "Take care of your coconut tree for 5 years, it will take care of you for the rest of your life."
A variety is a distinct, often intentionally bred subset of a species that will behave uniformly and predictably when grown in an environment to which it is adapted. Widest sense includes cultivar, ecotype, landrace, etc. Cultivar is a cultivated variety. Genotype means the hereditary constitution of an individual. Phenotype means the appearance of an organism with respect to a particular character or group of characters, physical, biochemical, or physiological, as a result of the interaction of its genotype and its environment. Inflorescence is the part of the plant that bears the flowers, including all its bracts, branches, and flowers, but excluding unmodified leaves. Coconut inflorescences have both male and female flowers.
On the left, you can see an Indian variety called Laccadives microtall. We see that on the same coconut tree, the size of the fruits is very variable from one bunch to another. The fruit size is often inversely proportional to the number of fruits in the bunch. The size of the fruits can also vary greatly depending on the nature of the pollen that fertilized it. The smallest nuts are often those resulting from self-pollination. On the left, the top photo shows a very productive Malaysian green dwarf, with an X-shaped canopy. A year later, the same coconut tree, photographed down from the same angle, looked completely different. Not only was the production lower, but the shape of its canopy changed and became circular. This alternative biennial production is quite common in dwarf coconut palms. In both cases, these are examples of seasonal phenotypic differences expressed by a genotype.
Allogamous designates a coconut palm that is naturally fertilized by pollen from another individual. Most of tall-type coconut cultivars are preferentially but not exclusively allogamous. Opposite, autogamous. Autogamous, reproducing naturally by self-fertilization, when the pollen of a coconut palm is fertilizing its own female flowers. Many dwarf-type coconut cultivars are preferentially but not exclusively autogamous. Inbreeding, producing offspring by self-fertilization or by crossing of parents that are very close genetically. Opposite, outbreeding. Selfing or self-pollination is possible for coconut palms because inflorescences have both female and male flowers. In tall coconut varieties, selfing generally produce an inbreeding depression, reducing yield and growth up to 30%. Hybrid. In the case of the coconut palm, the term hybrid is defined in its widest sense as the result of a cross between population, families, or individual palms belonging to two different varieties. The global area planted with coconut palms is about 12 million hectares. The global area of coconut genebank ex situ conservation is about only 1,000 hectares, so, about 0.008% of the total area. Most of coconut diversity is obviously managed and conserved by farmers. Scientists are trying, with limited means, to concentrate and conserve biodiversity in very small spaces, called coconut gene banks. Breeding and conservation in situ by farmers has created the extraordinary diversity of varieties and forms of today's coconut palm. However, farmers' selection methods are often ineffective in the short term. We will see why during this training. If scientists and farmers work together, a small improvement in farmers' practices can yield huge progress in coconut conservation. This is the main reason why we conceived this training. This slide schematizes the different components of the conservation of the coconut palm. There are ex situ conservation, either in the field or in the future by cryopreservation. Using cryopreservation techniques based on embryos, plumules or pollen carries certain constraints. It does not allow reproducing and thus multiplying a genotype, but only the progeny of this genotype. For most allogamous tall-type varieties conserved ex situ, embryos will have to be obtained by controlled pollination with bagging, and this will a very high cost. In situ conservation occupies a central place in the diagram. If scientists and farmers manage to work together, this will greatly increase the effectiveness of conservation. Finally, the polymotu concept, which consists in planting coconut palms in geographic and reproductive insulation, could serve as an alternative for conservation.
Dear colleagues and participants, thank you for your attention. I would like to finish this lecture with a thought to the late Dr. Ponce Batugal, my mentor and friend, who passed away recently. Over the past 20 years, his contribution to coconut palm research and development has been crucial.